Okay. Right. Okay. So welcome everyone again. So today we have the pleasure to have Professor Dieter Braun from LMU. To introduce Professor Braun, he did his PhD at the Max Planck Institute of Biochemistry. In 2000, he moved to the United States to work at the Rockefeller University with Albert Liebhaber.
Mm-hmm.
I have pronounced it correctly.
Yes, very good.
For 3 years. Then he returned to Germany in 2003 when he obtained an Emmy Nether Award to start up his own research group at the LMU in Munich. In 2007, he was appointed full professor for systems biophysics at the same university, at the LMU. And he is one of the initiators of the Origins of Life Initiative Munich. And then with the acronym OLIM. He has also been awarded several prizes and awards, including an ERC starting grant, a Klang Wilhelmy Weber Bank Prize. That probably went not that right.
No, it was good. It was good.
And more recently, he has also been awarded a Technology Transfer Prize from the German Physical Society. He has also founded the startup company Nanotempe, and he has received the Deutsche Gründerpreis for it. So thank you very much, Dieter, for being with us today. And whenever you're ready, you can.
Yes.
Can
Thanks very much for having me. Thanks for everyone. Still see a bit folks in the audience, and I'll try to make the talk really in 45 minutes, and then we have ample time for questions afterwards. Okay. So our focus is trying to figure out how life emerged. Basically, that sounds a bit high taken, and I'll just guide you along the logic and the ideas we try to make this happen and try to make progress here. As you see in the title, we want to have non-equilibrium settings, and the Earth was shattered by the moon forming impact, and but somehow managed to have these crazy delicate molecules like RNA and DNA keeping the sequence over 4 billions of years. The universe only is 13 billion years old, so half of that. Well, the information could be preceded, Darwinian evolution improved, and life could survive on Earth. That some lucky things on early Earth, but the question is a bit, because as you go back for evolution, and evolution gets always faster and faster, is it so fast that you could actually do lab experiments to figure out how these first steps, first sequences would emerge and sustain themselves? But the point with the non-equilibrium setting I want to make is that the Earth must have provided some non-equilibrium to make this replication happen, to make life happen, because life is non-equilibrium. Nowadays, it's in a cell, it can feed itself. I think initially the early Earth must have provided some of that initial energy. And if you look how that could be cooking lakes on Iceland, or hot vapor coming by, porous rock after a volcanic eruption, or. We will show a little bit that we're more interested also in air-water interfaces. So if you go very deep in the ocean, it might not happen the way we think, because there's no free gas anymore. It's also dissolved in liquid, but higher up, you'll get that. And basically that's a typical rock you pick up on Iceland or any volcanic island, has this porous structure. Okay, fair enough. Has these porous structure, and the idea is a bit, can these pore host the first molecules? And what we've explored after a while, where we only looked what processes in the liquid, is that if we have air bubbles or in vicinity to the interface, that might be an interesting setting. And for now, we've mostly focused on a temperature difference, if you would apply that to that pore, and that's what we do experimentally. And the very first thing you see in such a setting, if you have water here, gas up here, and for example here, a fluorescently labeled DNA molecule, that within 100 seconds, it accumulates at this boundary here. You see that bright line, because water, if you subject the setting in the temperature gradient, will continuously evaporate, take the DNA with it, and only by diffusion it can hinder itself to be, really located up there, and water, gets the water vapor gets in the cold side into bubbles, and a lot of things are happening at the interface. And we just figured out that the interface is quite active and is doing things which RNA world or RNA molecules actually would love to have. So that's our focus since, say 3-4 years now. And I want to show you some unpublished work and where we are at this stage and how we could think that that could be actually a crucial boundary. So that evaporation here, you can actually measure that fine line. You can get measurements on the concentration. So 100 mR of RNA DNA gets accumulated about 200-fold. And the line you see here is actually a calculation from including all the physics of the interface, evaporation dynamics, everything into engineering program, which does a finite element analysis. And that helps us a lot to, explore, other conditions before we do the experiment, simulate in the computer what do we have. For example, this accumulation is not limited only to the DNA and RNA, also salts will accumulate 5-10-fold, and we'll come back to that. So we've seen in that publication that a number of processes are happening there. For example, if you have an air bubble and have your molecule already at quite high concentration, it tends to accumulate at the round contact point of that bubble in the water, because again, evaporation, recondensation is going on, and you can form crystals. And those crystals you can actually put in X-ray crystallography machine. It's collaboration with Matt Pauner here, and that's not just some molecule. It's the molecule which is considered to be intermediate molecule before creating RNA, and which actually has the interesting advantage that it nicely crystallizes. It actually also would be chirally selective in the crystallization. But you also see things that as you accumulate things at this boundary, you reduce the volume of water as you do that, and you have a fluctuation of that interface. So if the water goes down, like on the shoreline, you have low tide, kind of on that micrometer scale tide interface, it actually dries down the reaction, and you have a dry, in this case, nucleoside at the interface, and then droplets will fall into it, so it goes up and down. You get wet-dry cycles, which are tightly connected to these bulk of water, therefore, always being at the interesting or defined salt and pH concentration. And what you find here, for example, is that can phosphorylate the nucleotide by just having, phosphate in the solution. So the setting could activate those molecules, but then if you have ribozymes, in this case, it's a very simple ribozyme which can cut another ribozyme. It's recorded by fluorescence. If you cut it, the dye is not quenched anymore by the star on the other side, and it lightens up. And what this movie actually shows you is that it all happens at the interface. So at the interface, the concentrations of these parts are higher. Also the salt, especially magnesium concentration, is higher. And then the whole thing is driven at the interface. So ribozymes, the main part of RNA world idea, really like these interfaces and are really promoted by the interfaces. We'll come back to that. But the interface, if you have, a specially designed sequence, which likes to bind a lot to itself, also to itself that it forms a network, is creating these, dragon-shaped snakes, which are, almost millimeter sized, because, first the single molecules of the sequence are attached, but then they bind together. Diffusion constant drops. So you have a runaway accumulation process that these things want to bind together until, as you've seen in the movie, they detach from the interface again and get back into the convection flow. Again, for orientation, this is the water phase. You see up and down movement because you heat the front and cool the back. And this is an air bubble. And at the air bubble, the interesting things are happening here. And of course, that's interesting. If you have double-stranded, large-scale things, aggregates, it can survive longer because the hydrolysis, RNA tends to be broken up by water, is much lower if it's double-stranded than single-stranded. So it would be an interesting storage of nucleotides, oligonucleotides. But then if you have lipids and the DNA, two-color imaging here, both are accumulating. You accumulate the DNA on the right channel. You also accumulate the lipid, and the limit up here in these complex dynamics of the bubbles here is actually forming, what looks like cellular structures. And they're rather complex. They float around, and actually they sometimes get larger and larger and can be ripped apart by these Marangoni flows up here. And you can actually by that get, cellular division. And, this is a complex thing. It is likely multi-laminar lipids, and the DNA inside. What we could see by fluorescence is that if you want to destroy the RNA inside by proteins from the outside, it's not destructive because it's really encapsulated. And actually the environment inside is, physiological. You can have an aptamer, RNA aptamer, which apparently folds nicely and captures things and is happy inside this. So, that would be a bit the idea, that you wouldn't start with cells. You would start with these pores, that things are confined, running around the pore. But eventually these processes could accumulate and encapsulate part of it into a spaceship, which can go to other places. And that by also trigger the emergence of cellular systems on the way. Okay, but this is all a little bit in the periphery of what we are interested in origin of life field, right? We would be really very much interested to go for the replication RNA thing, and that we only tackled recently. So what the interface can give you with this accumulation, that's a bit the idea. The accumulation gives you a higher yield because even a slow reaction in the bulk, for example, would be accumulated, and therefore you get high concentration of the reaction product at these interfaces. So we could tolerate molecules which are not super active. And we'd like to use that for using a chemistry which is a little bit more on the recycling side of things, so that molecules are activated, recycled, go back and forth between these states. And we probed a number of these ways to activate the nucleotides and make them polymerize, basically. And they're very harsh chemical drivers, like EDC in this case, where you get a lot of side products. It's very fast, but it's almost, we tried to do a ligation chain reaction. It does a lot of other things on the sides. Imidazole is what Jack Shostak is pushing for replication on strands and template and get base by base addition. Really wonderful work, figured out basically all the details of that reaction. Still, it might be a little bit complex to make it. They have scenarios to activate those in situ and get it going. And then what we are now focusing to make that activation chemistry more simple, even more simple. That's a bit the idea, just go with phosphate. So we did a lot of work together also with Judith Bono and Bruno with a 3,5' cyclic, which can polymerize G, but G only so far as we can see. And it's actually a little bit difficult to get this phosphorylation here. It has a lot of energy, it has a lot of strain, but to make it prebiotically, you have a little bit trouble to get that. And then we focused recently on this one, which initially we thought everything was done in the 70s, but it turned out that in the 70s they did a lot of work with AMP, cyclic AMP, and not GMP. And that was kind of the key on what we will tell you today. So because what this reaction in our hands was capable of is doing a number of these steps, at a little bit elevated pH. You can't elevate it too hard because then you break up the RNA by hydrolysis. But what I think was crucial in this expertise here is that you can do it at low magnesium concentrations. Why is that important? If you have high magnesium concentration, RNA is super sticky, double-stranded form. It's very hard to get it apart and make those replication steps where you first make the a single strand, make a double strand, get it apart, repeat, and go for replication cycles. And that's especially the case for this imidazole chemistry, which kind of runs at 50 millimolar magnesium. And then you have really a hard time to get that matching up with this. So the second thing why this is interesting is that in the dry state, it is actually possible to make this phosphorylation. And papers in the 70s actually complain about it because they wanted to have the phosphorylation on the 5'. They always get this cyclic 2, 3' phosphorylation. So that's actually rather straightforward thing to do. And what we figured out that they actually also polymerize. Not yet to the point where we'd love to have it. I'll show you the details, but quite a bit with the hope that if we could improve that a bit more, we could get those strands to actually hybridize and then trigger, for example, you have here a strand with the 2, 3' end still there, or you recycle that by the phosphorylation chemistry and another one and go for a ligation reaction. And that's a reaction which is also in the the record for 10 years, and sometimes works, it doesn't. So, but pushing for it, it seems to be actually quite a reliable thing. And then it would be, of course, if such a setting could be capable to, under these conditions, separate the strands, you could repeat that and go for very slow initially, replications, with the hope that that system could find a shorter sequence, not these large ribozymes of 200 bases. That might be a long stretch to find those, but perhaps we are lucky to make shorter ones and get that going and get to real RNA world in the lab just with this setting. That's the dream. Okay, I'll show you where. But we are actually a little bit on the optimistic side that this might actually be possible. Let's have a look. So what is part of the story is that if you go for hydrolysis of these strands, if at these elevated pHs, the strands break up by the addition of water, those to a good extent are already 2,3'. But the idea is that you keep that going under condition with a number of phosphate molecules or activated phosphate molecules to recycle those. So the reaction will cook and cook and cook with the hope to get interesting ribozymes out and interesting evolution. So let's see. So we analyze first the polymerization of this. And that's first you need to do the homework. How do we detect those? On the one hand side, we need HPLC, which selects the length of these molecules by, chromatography, just basically detecting how much free phosphate you have, and that determines it. We do run a precipitation protocol, which I explicitly mentioned here, which is optimized because it goes overnight to keep also the small concentrations, dimers, trimers in the mixture. I think that's also enhanced by the fact we have not so much high salt to start with. It has an ionization fingerprint, which is dependent on length, and we correct for that. And we have checked for the linearity of these in our AZTOF, standard mass spectrometer machine over 3, perhaps 4 orders of magnitude. It saturates a bit if you go higher because it can't keep up with the counts, but it is quite a nice instrument, and we worked a lot on that. So how can we get the most of our instrument? For us, that's actually, and that was my job, to make a LabVIEW program, which actually loads the real raw data from the AZTOF. And which creates one of these spectra, which you can see here more or less every second. So it's tons of data and really very nice data to know what it is. So these larger peaks, in this case, are already polymers of the G. And then you get these triangles, which, in any of these AZ, you get co-flying species which have ions attached to it. But these can be calculated. It can nicely identify and can really know, okay, that's the mass, I understand it. So the overall idea is that you get these mass spectra, you fit them actually with your model of whatever 5,000 masses you're expecting, and hope to get a good coverage of it and a good fit of it. And that's what we do. Basically, we get the isotope patterns of each of the species, which has number of peaks depending on which molecules are inside, and fit that to the spectra. If we have, only poly G or GC combinations of sequences, that's good. They're far apart. You'll get those easily out. But if they get close, as you go for all 4 bases and 5, 6 mers, you have to actually do a fit, which is overlaying these isotope patterns and add them together. So in yellow is the isotope pattern of one sequence, and then there are others which together give the sum of that red overall spectrum, and that's fitted to it. So we really try to get most out of it, and it's actually fascinating how much the machine can deliver. Okay, so if you do that, and that's work of Avinash, Das, and Sri and Juliet, and kind of the chemists in our lab, we did a number of checks. And one check is that it needs a bit initial pH of 10. It runs without salt. It likes to have a little bit potassium with it, which might indicate some quadruplex structures. And it doesn't like low pH. So we have to be, 8 to 12. At 12, you run a little bit much into hydrolysis. We can do NMR. We can see the linkage in NMR, but we need a lot of material for that. But we might follow up on that in the future a bit more. and it runs at low temperature. So basically in the dry state where this proceeds, so you dry it, you wait that it reacts in the dry state. 40 degrees is quite a good number. It drops a lot to 30 degrees, so that's interesting. And you can even get 60, 70 degrees, something going. You see here the salt dependence. Sodium is still okay. If you add 2 equivalents of that, magnesium is kind of detrimental. In this case, you would have 40 millimolar magnesium. That kind of kills it. And that's a bit understandable because you destroy the initial activation you had inside before. And that's actually good news because we'll be able to separate the strands likely, well, if they come together. So this polymerizes a lot for poly G. It does a little bit more for U, but G is kind of the driver. And the others don't do so much. But the interesting point, and that's the push we want to go in the future, is that if you do mixtures of it, can the mixture, which would have G and G would actually polymerize, that polymerized structure, can that be used as a matrix to co-polymerize the other bases so that everything gets polymerized together? And that works a little bit for GC. We get, trimers with the full coverage, but, this logarithmic scale, those are much less concentrated. And you get, the longer ones, kind of, you have G and then some C inside. We don't know where it is, but that's something what happens. And the gratifying point here was the following experiment done by Gelate, that we have cold side and the warm side. And you have this accumulation, as I showed you before, and that's kind of a white light image where you see a lot of bubble dynamics on top, and this is the bulk at the bottom. And actually that results in these blue dots, and the blue dots actually have consistently higher concentration than if you would just dry it down. So this recycling actually works, and it works quite well. And we get longer structures. And this is a run for 5 millimolar of the 4 bases. And, you get formers, but not all the sequences and formers. That's the aim. Can we, optimize that, run it longer than that 1 day? We're restricted right now. Can we go better settings, understand it better to actually get a little bit longer and all the coverage? And one idea for that, to get those high concentrations, is to go back to an idea which we explored before, which is a thermophoretic thermogravitational trap. So what is that? It basically takes the warm and the cold side with quite a strong gradient, where you have, let's say 60 degrees here and 30 degrees here. That triggers a thermal convection, but at the same time, the molecules go from the warm to the cold side with an effect called thermophoresis. Molecules move in a temperature gradient. That's actually the basis for that startup. Well, it's not a startup anymore because they have 150 people working for them, nanotemper, because that can be used to detect binding of biomolecules. So sometimes you make the bridge from origin of life to biotech. So that's always something to say at this point. So that accumulates the molecules actually quite effectively. And we've managed to get that going in the lab. Initially, we had experiments with lasers. Now we do it really with basically a thermal gradient only. And that's all water, right? All the process I showed you until now is that we want to have it in the dry state. It turns out that this interface dynamics of water and air can be that the air is on top, but since it's run and working mostly by surface tension, you can also have the bubble at the end. And Christina Dirschel found out a way to make that thermal trapping and the droplet at the bottom to go for the polymerization. And typically what's happening in here, as I said, it's this convection flow, very laminar, and at the same time you accumulate by thermophoresis your molecules. This is here both visualized by fluorescence. And that's done in a format where our simulated rock pore is really made very well defined, kind of all the physics have sapphire windows, that we have UV possibilities, but also that it's just very well characterized. You see, it's a very nice convection. These are Teflon sheets, kind of how you define the experiments much. And these are now really well working. They run for weeks, no leakage. That's due to Christoph Mast in the lab, really an expert and great help in designing actually these chambers and engineering it. Just as a side remark, that's work by Noël Martin, which we will hopefully write up soon. where actually you can make now the leap from origin of life RNA to the first living cells. These are cell-free extracts, if you know what that is. Basically extract all the goodies from the cell to replicate, well, also to replicate. You know, Hans Mutscher is doing that in a very good way to have expression systems from RNA, from DNA to RNA to protein. And you can actually see that those proteins are also nicely accumulating, actually to the point that if you dilute it and actually destroy it, because it's not expressing in diluted state, the trap is getting the molecules together, and then it expresses here the GFP molecule. And that's, I think, quite fascinating that even, you know, well-developed life would work in these pore systems. One question we are asked quite often in these, you know, as I said, it's 100, 200 micrometer across. We make several degrees across it. It's a very steep gradient. You know, that might be really localized to very specific places. We can make the argument that that still works quite well if you have less steep gradient, if you tilt the traps to some angle. And that's just, again, simulation calculations. We are not doing these, you know, gradients over 30 centimeters because it slows down the accumulation. And we just want to get as fast as possible in the lab. But it looks as if these pore systems would be still capable of doing the same thing, but a little bit on a slower time scale. Okay, back to the polymerization. What's happening here? Well, first we can check how much we are accumulating these nucleotides, because the idea is you feed them at rather low concentration on top, micromolar perhaps, and then it should actually accumulate exponentially. That's the expectation if you calculate it in a computer. And that's also something what Christina was seeing in the experiment. They really accumulate to millimolar levels. This is a steep logarithmic scale down here, and these are experimental points, you know, tested in the AZTOF with a simulation line of the whole system. So basically, this warrants that if we would have very low concentrations, you could actually accumulate for the polymerization, and the polymerization and that dynamics might take time. Okay, these are simulations. You know, you see the timeline here, up to 10 years to get phase. And Christina was finding that the polymerization is actually quite a lot. And Christina found that the recording is actually quite a bit better in these trapped systems, to the point that you get more coverage of the GC sequence space in it. And you know, while that is very important to make the double-stranded form eventually, it is also illustrating the point that you can couple the polymerization, tripolymerization, with this very strong accumulation mechanism to actually keep molecules in these rock pore systems together. Okay, back to how to get that 2,3' cyclic phosphates. Tri-state polymerizations are out there to make that. One case we have seen, and where this works reasonably well, is again similar pH, a little bit higher temperatures for this one. We look for 3 days, it might be faster, let's have a look. And our guanosine, which is the molecule without the phosphate, gets transformed quite a bit to having 2 and 3' phosphates, which are not yet cyclic, but either or, but quite also a number of them which are cyclic. And what's important, not get much of the 5' up there, which you want to link to. So it's important that, you know, there's 5' is this location, that you don't get the phosphate in that one. And lo and behold, if you now look for the analysis of this molecule, you already see dimers, although you actually only wanted to look out for phosphorylation. So the coupling from phosphorylation to the polymer seems to work under these conditions already a little bit. Of course, that's a dry state, it's not yet in the trap, and so on and so forth. That needs to be a lot of more optimizations and analysis. But it's nice to see, because this trimetaphosphate, this circular phosphate, is something you can think of getting also in these traps by a little bit higher temperatures, and if you have phosphate and cook it long enough, more or less, you can get that. But that's up to see. Now, if the polymerization, right, is good enough and they can bind together, hopefully they bind more, or that's the expectation if you accumulate them, because the salts come to there, also the concentrations are higher. What would happen? Do we get, if you have a template and you have one strand which really gets here to the end with a cyclic phosphate end here and another primer, would you get that ligate? And that's been published in the 10, 15 years work on that, where this molecule was only prepared by hydrolysis. There were also from Braun experiments where ligation were seen, and it was not clear what was going on. It's likely that they produced a cyclic one, that's at least our interpretation at this point, by hydrolysis, and then could move around the structures because it's C and G only, and then make the ligation reaction. But, you know, Holliger and Phil Holliger, Hans Mutschler has looked in that in ice systems, and John Sutherland also looked for this reaction in bulk, and we also tested it, and you know, it reasonably works. So we would have an escape route from the phosphorylation towards a replication cycle. It's slow, needs days. You know, even can go for these hairpin cases. If you remove the primer too, you see actually that these hairpins also ligate together. So the hope is that if we can separate those strands again and run this long enough, that we could really look forward for replication reaction. Okay, I need to go a little bit faster. Well, forward. There comes the work of Alan Ianniselli, who was checking out what are the processes here to separate the strands and how can we help in doing that. That's again, you know, saying that how these on the warm side droplets, as they dry up, you know, make coffee green effects. It accumulates at the boundary mostly. But what the evaporation gives you, even if you have a very salty mixture, is that the evaporated water is, of course, pure water. So that pure water gets in touch with this. And then, you know, initially, because they, of course, also dried up salts, initially you'll have a very low salt concentration. That helps already a lot to separate the strands. But there's a second point to that, that if you have, and you see here this nice drying dynamics at the interface, you see the lighting up with fluorescence, although dry fluorescence is very much lower in brightness. If you do that in a CO2 buffer, it adds another story to it, that water is very low in pH, and low pH, like 4, is actually helping also to get the strands apart. It's a nice publication, so Sutherland on that, and it's actually, yeah, it's just based on the charges of the RNA molecules. And Alan could monitor that. He had a fret where he could look, if I add now the CO2, everything gets blue and the strands are separating, and that's co-illuminated with a pH dye, and you could see in the pH dye that the pH drops. So we could get the full characteristics of how low salt, low pH is helping to separate the strands. And what we typically do, if we think we have a condition which could replicate things, is go for polymerized chain reaction. So we add a protein, kind of make a big jump in time for later conditions, very efficient way to replicate things. And it was, you know, initially you start with 100 bases, and you want to figure out, is this reaction under these conditions giving us longer pieces, right? And what he could figure out that it actually does so, while in the test tube, these reactions typically make shorter and shorter pieces because they replicate faster and the competition for that is lower. So the idea is that these accumulations, which are length dependent, are actually promoting longer strands. And, you know, this is a reaction, if you have a cyber green dye, which illuminates things by fluorescence, you see these bubble dynamics, of course, sped up here, and eventually you see a big pile up of material of double-stranded DNA in this case. PCR runs in DNA, but you know, all the physics very similar to RNA and DNA. Okay, so just give you an impression what's going on here at our microscopic shoreline. Interesting point here was that if we run this at temperatures where you could actually not separate the strands, if you do that, you know, just in the cycle at these low temperatures, you're not getting anything. You also don't get anything in the chambers if you're not adding CO2 to it. So the CO2 makes that pH drop, and that was actually really necessary. Now, everybody knows that early Earth conditions would have had quite a high number of CO2. So that's quite realistic for prebiotic conditions. Actually helps the replication here. Now, the interesting point here is that we can analyze the sequences. We get really long sequences, but they had an interesting fingerprint of seeing mostly AT sequences. Although we have GC-AT balanced out in the beginning, we see more of those AT. So the reaction really wants to go for longer strands. And since AT melts easier, it was actually biasing the sequence to potentially really separate the strands. But it was unclear, you know, does these conditions actually give us a chance to separate 1,000 mers, right? And the calibration Alan was doing here was actually capable to figure out how low pH, low salt conditions in the chamber would act. And lo and behold, he got a phase space image of that. He knows, you know, what's melting temperature depending on these conditions, and he imaged it in the chamber so he could extrapolate what to expect. And the interesting, and we didn't believe it initially because it was too good to be true. Basically, if you look for sequence count, GC and AT, sorry, that's a bit cut out here, it falls on this line from the sequencing. And this is exactly the line you would expect when the strands are actually able to melt. So what you actually find is that you start here. These can't really replicate as well because they can't separate enough because they have too high GC content. But then they move to this line and live there, actually get longer. So our short message here is basically, as we looking in these water-air channel chambers, and if we would have a very efficient replication mechanism like for the PCR, it would run you to longer strands, would give you quite good separation capabilities. That makes us optimistic because now we want to go for that replication to do that with ribozymes, and show some preliminary results here. Again, the hammerhead ribozyme is happy at the interface. We collaborate with Hannes Mutscher here to actually dig out an old ribozyme molecule, SUN-Y, which is able to ligate. It ligates by cutting off the G and then do on the template things together. So here we assemble this hammerhead ribozyme you just saw active in the final form in the second before from 3 pieces by this large SUN-Y molecule. But typically the problem is you can do that, right? You have everything on the template, but you'll never get it off. So that's also what, and Jack Shostak studied that molecule way before. What people figured out, this molecule needs 50 mol of magnesium, is happy. It makes the hammerhead ribozyme, but you'll never get it off the substrate. You get it off in a gel because the gel is made to separate the strands, but in situ you won't get it off. So you won't get active results of that ribozyme because it's stuck on the template. So what the Fock chamber here offers is that the SUN-Y can make it at very low magnesium concentrations, 5 millimolar, even 1 millimolar is possible. And of course, the question was, is it active inside the chamber? And this nice blue line here actually shows that. So after 4 hours, already a little bit after 2 hours, we see the substrate on which the hammerhead works to cleave it really be active. Now, of course, if you want to dream, right, the experiment is to figure out 3 small pieces to make the SUN-Y itself, by itself, and hopefully cleave off that substrate. And that's what Annalena Saudit is doing with hard work with Leonie to get this together and get already all the 3 parts together, which actually assemble in SUN-Y. And the big 100 million question is that SUN-Y active? And can we replenish the replication by itself? So that's kind of an R-enabled scenario, which, SUN-Y is 200 bases, that's assuming that you have all the sequences ready. We, of course, hope that similar things would work also with shorter ones in the dry state, things like that. So a little bit short mention on chirality. There are some ideas on, because G is pushing the polymerization here, that you get that possibility. And because you have some non-linearity here, is that if you have right-left mixtures, that the polymerization would actually then prefer the initially higher concentration of those 2. And we like it a bit because G is kind of the dominating part in the polymerizations, that it would select the chirality by the G, and if those co-polymerization would happen, it would also prefer the right chirality for the other bases. So there might be some chirally amplification mechanism in these polymerization settings here. Okay, those are people doing the hard work. Was great fun, and is great fun to, get them on that hypothesis and make it go. I think I should come to an end here. That's our scenario, that we have a little bit more time for questions. These are other stories. You can have a look at the papers. I would have slides prepared if one wants to go in the detail also later on. On the one hand side, it's length-dependent PCR, similar than the air-water interface, but we had it only without air bubbles, already selective for length and replicating the longer better. Annalena worked together with Jerry Joyce to make one of his Ribo PCR work in the thermal convection that you can read out in the PRL from last year. So we could actually also accumulate the molecule and have it, work as good as in the test tube thermal cycling, just by having a hotspot. Here we looked for ligation. Now, not as shown before with this 2,3', but with a ligase, with a protein to be fast. But we figured out that if you have random initial molecules, what you get out, initial random sequences, molecules, you won't get random sequences out. That's actually quite fascinating story. What are the selection pressures in ligation chain reactions? Not giving you the full randomness. And for evolution, that's good because your sequence space is getting reduced by these processes, and you likely have a higher chance to jump into Darwinian evolution. And that's a funny story on how tRNA can be made into a replicator, but that's a long story in the life. And just to give you an impression how it looks in the lab. We're happy enough that we could initiate collaborations with the astrophysics and particle physics. It gives us crucial funding for instruments at Munich, but we are right now applying for the second 4 years of this Origin of Life Collaborative Research Center, CRC, from the DFG, German Science Foundation. And keep fingers crossed, decision will be in January. We have compiled 400 pages and we'll push for the next round. So the good thing on this, kind of the first time on that scale that Origin of Life was funded, and we could get another 4 years and another 4 years if we're lucky. So let's hope for the best. I'm part of the Science Collaboration Origins of Life worldwide network for the Science Foundation, which unfortunately, ends after 10 years of very solid funding of this collaboration in 2023. So we need to think how to go after that and make people interested in the topic. And with that slide, I want to end. And funding from ERC was very helpful, and like to ask for questions. Okay, I'm just, I can stop the share and we can come back to it in a second. I hope you can still hear me. Raise your hands. I can see my own image. That's okay. Yes, I see just in the chat. Okay. I mean, please, either you grab a microphone and get in touch with me, or put up questions in the chat. I'm happy to follow up any of those.
I have one question, and it's related to the size, the importance of the size of the pores in your reactions, and the kind of rocks and environments that would facilitate those reactions.
Yeah. What's the question? I just tried to go back to that slide, but so
essentially, is there an optimal size, or which kind of rocks would facilitate this due to the size of the pores? These reactions at best. Yeah.
So there are two mechanisms. The one runs on thermophoresis, that's quite size selective. So you need those 150 micrometers. If you tilt it, you go up for 1 millimeter about, and then you can have that, elongation of the water, of the gradient in your rock, but you slow the reaction down. Eventually it will accumulate the same level if you make that long enough, but it will just take time to get it. If you do it too large, then basically convection gets too fast, and the time for that thermophoresis equilibrium against diffusion to equilibrate will not work. Then you just get convection. You just do thermal cycling, nothing else. If you get too slow, too much together, convection is too slow, and you rather get that small thermophoretic movement, which is probably 50% from warm to cold, and you also get no accumulation. So that needs a special size. But as you know, studied here, you know, if some of the widths are not correct, like here, diffusion will just cross it, and it will, you know, focus on these thicknesses where it's actually optimum, and that those will accumulate. So in a porous system, it will take more time, but the prediction is that it will still accumulate, perhaps not as much as in our best case lab experiments, but still quite significant. And I tried to, you know, had these slides, which, you know, basically show for optimal cases 2 centimeters to get 5 orders of magnitude of accumulation. So we have some leeway there. That said, that's only a thermophoretic trap. If you have air-water interfaces, that's much more possibilities. You know, if you go for 500 micrometer, it's quite nice dynamics because the droplets grow and actually touch then the other side and then fall down, and you have a lot more dynamics. If you make that larger, it's getting a bit slower, but the accumulation is still there. So there's much more relaxed to get similar accumulation and melting dynamics.
Okay, yeah, thanks.
More questions? I mean, we've been asked that you know, is it realistic or not, time to time.
Yeah, can you hear me now?
Yes.
Yeah,
There's some
so we have a
feedback, but
couple of
otherwise,
questions. Yeah.
Yes, please. Yes. Just continue again. I'm not hearing you.
Neither do we, yeah.
Now it's good. Now it's good. Just continue. It's good. I can hear you. No, wrong person.
May I ask you something, Dieter?
Yes, please. Yes.
Okay, I am Carlos Briones from the Molecular Evolution department here at Center for Astrobiology, working with RNA also on molecular evolution, and you know. And first, I would like to thank you for this extraordinary talk. Such a nice amount of work you have been doing for years.
Thanks.
So congrats. Congrats. Yeah, and I have a question regarding the regioselectivity of the polymers you obtain. I mean, how do you measure that the amount of 5' 2' in particular links are avoided? Because so I mean, it's very interesting to use this cycle phosphate 2', 3', of course, as you have been explained very well. But it's easier to get the wrong polymer in the sense that the possibility to bind from 5' to 2' instead.
So we are running some digestions, and at this point we kind of got conflicting results. That's why you don't see anything in it. It is likely that we get a lot of the biologically wrong 2 5', but it's at this point hard to quantify. If the quadruplex is really the driver, we might actually get more of the 2 5'. You know what usually the argument is, because the hydrolysis of the 2 5 is much faster than 3 5. You know, eventually in this recycling way, you would select out the good ones or the more tighter binding and so on and so forth. We have not yet figured out you know, how that ligation reaction would still work if we have a lot of wrong linkages in it. It might, you know, still be sequence selective and wouldn't care about it. So there are a lot of questions to figure that out. You know, unfortunately, the mass spec, of course, doesn't distinguish. And at this point, we have to run through, you know, proteins which have some tricky time and very special characteristics in these mixtures, so it might be take a little bit more time to really figure that out. The NMR, phosphorus NMR, also not gives us more input there. So we would tend to just ignore it a bit for now. Just say its selection might take care of that. there's one escape route where we can either go for pore sequencing, where we would, if things are long enough, wouldn't care about the linkage, and could compare that against Illumina sequencing, where the pre-preparation to DNA would actually likely reduce that linkage, and that would give us a handle to experimentally explore it. But otherwise, it could even be that the thing I was sketching to you could even tolerate quite a high number of the wrong linkages. So that's where we are at this point. Honestly, it's just experimentally a bit a tough thing to do.
Yeah, you're right. It's quite tough, in fact. Yeah, and I have a second question regarding the maximum length of the polymers you get. I mean, it's 10 nucleotides long?
Yeah, roughly. Yeah.
So
I mean, you know, the AC ionization goes down also a bit, then we might have a little bit more. But
so 10 mers is quite good. But could you envisage any functional implication of these relatively short polymers? I mean, could any ribozyme activity rise with this short length? I mean, ligase or something like that? Because, I mean, in particular,
very good.
Yeah, because I'm thinking, because we published some years ago a model for increasing the complexity of RNA through ligation of random RNA oligomers. That was an in silico model that was used, by the way, by Holliger et al. to envisage some of the experiments they could do. And okay, the minimum length that in our simulations was required was about 35 in order to have ligase, an RNA ligase that could originate a kind of stepwise evolution based on ligation, you know. But, so could you envisage something similar with only 10 mers? I mean, stem loops and nothing else. I think it's a tough question.
I'm trying to persuade, you know, Jerry Joyce and the like, that 2,3' ending could be an interesting target. You know, right now everything is in D and TP endings, and then you get these large. But he already suggested some hairpin ribozymes, which might be shorter, you know, still on the order of what you say, right? I mean, there might be one way, if we really go for ribozymes which work in these dry state scenarios, you know, that you would have a ligation state and then go in the dry state and then help. That could be smaller, shorter. That's the hope. Otherwise, we might need to wait for some of those ligation steps to get you slowly through to make it for 30 mers. You know, I think it has to be. There had to be finding these shorter intermediates, because the sequence space is much too large for the longer ones. Yeah, so you wouldn't be able to select that out. But of course, the hope is a bit that we could identify them by experiment, then figure out whether the trap, if we would run the trap setting, if it recycles nicely, you know, for weeks or months, that you might rob your way already in these shorter timescales towards that. A lot of interesting work to be done. I think, you know, they're good starting points if we get the polymerization a little bit better, and there might be a real demonstration on everything, which would be, of course, great, because you could say, look, these are the steps of how Darwinian evolution in such a setting could lift off the ground. The big hope is that these small ones are doing already a little bit. You know, the advantage is that you have the ligation already operating slow, but a little bit. You know, that's always a good starting point for looking out for improvements. So you know, you're not having a big gap on which some magic has to happen to jump on the other side, but you have some slow process which already gets you in principle on the other side.
Yeah, I fully agree with you. So,
you know, amino acids could help, you know, whatever you think could be around, and who knows, right?
Peptides, perhaps somewhere there helping in the polymerization, abiotically originated peptides, I mean, so could be also involved in the soup. So
absolutely,
yeah. So,
you know, with all the progress of Matt Powner, and you know, that could be tried to link that together, and then
the systems.
Phil Holliger had these K10 amino acids which are helping. So all that, you know, some possibilities needs to be considered. Right now, we are happy that we can do it kind of really minimal. You only need G kind of to start, and then slowly the others could come to it. You might be able to run the system on GC only or things like that. So just to minimize the amount of molecules you need to go, because then it would be, I think, more realistic probability would be just much lower than if you really need that complexity. So I still hope that if we would only start with GC, find a good sequence who's doing that and not needing anything else would be actually my preferred mode, because the probability of the whole scenario is just lower. Higher, sorry, yes.
Okay, so thank you again for your talk and the explanations. Thank you.
Hi, Dieter, thank you for your talk. It's very informative and we've learned a lot. I just want a single question. It seems that the key of all these processes, the 2,3 cyclic or phosphocyclic GMP or nucleotides. The point is, which geological early Earth scenario you envision for this type of product? Because phosphorus is not the most common element in the early Earth.
Yes, I mean, there's progress. You know, we also run experiments where we, you know, leach out phosphorus, and you have to talk to Matthew Pasek as an expert. But you know, the whole idea that after the moon forming impact, and you have for quite some time also more reducing conditions, also more capabilities, how lightning could release phosphorus. Let's have a look. But yes, I agree. You know, these scenarios must have been localized somewhere where we have ample sources of phosphate, and that might be, you know, the chance part on the story, because that would not be distributed on the whole Earth in a similar way. And so, but also getting the G right is still a bit the question, and how could we link our scenario together with a setting which could produce us that from even more simpler molecules, or you know, that's still a little bit an open question. You know, John Sutherland synthesis works well for pyrimidines, not so much for purines. There might be different purines in the process. We might initially not have G really, but you know, that we just neglect a bit, right? And just say, okay, let's assume we have that G molecule and go from there. Let's analyze that. But perhaps if we have that scenario, we find also ways how the making of the G could be enhanced. That would be, of course, the great. Well, that would be the expectation, but it would be great fun if that would work out. You know, that we really converge to a scenario.
Okay, thank you.
Okay, I suppose that you hear me.
Yes.
Finally, I can make my question.
Yes.
Thank you very much for the talk. I think it's quite interesting the way you really go and increase the complexity and even keep the complexity. I was looking to your talk from an astronomer perspective.
Yes.
And, you know, I mean, in astronomy now, I mean, astrochemistry has been sort of successful to explain the formation of a number of molecules. But we get in more and more complex stuff in the interstellar medium. So my question is, I mean, you showed a nice process where you need to have liquid water. We know that at some conditions in interstellar medium, we can have, you know, the water in these porous to be liquid. My question is about the conditions. I mean, not only the sizes that Paco asked you, it's about the conditions. I mean, you're talking about temperatures which are always very similar to the ones we have on Earth. How much you can, you know, extend your physical conditions and still your scheme be able to work? I mean, in terms of temperatures and pressure.
Okay, I mean, the pressure for CO2, that would be quite flexible. We can run that, you know, as low as 100 millibars. It would give reasonably similar results. You could push it for 50 bars. You would get the same, you know, good separation. Temperatures, the polymerization can work in the dry state at 80 degrees. I'm just a little bit skeptical, because at 80 degrees that the strands you're making would actually make the double-stranded conformation, which you eventually have to go for replication, won't be 80 degrees. That would be rather those 10, 20, 30, 40 degrees. Unless you have very high amount of salts, you know, by that you could move it a bit up, so that 70 degrees you might be able to do, but those high temperatures just decompose RNA much faster. So any colder area will have a big advantage. So you could put numbers on these processes, and you know, how to make the G, you know, that's, I think meteorites are still very informative, not so much that they would bring the material, but they report to us what other bodies in the solar system was capable of doing and making. Bases are possible. Nucleosides have been found, very low amount. So you know, it's not unreasonable that these molecules can come about, but of course you'd like to have that as a real system in the lab to really make the whole stretch. Let's see how far we can go there. Hope that answers the question.
Yeah. Yes. So I have another question. I wrote it in the chat because we had several problems yet with the microphone, but I can.
Sorry, I didn't. Yeah.
I can ask you now that is working. So this is also a question from an astronomer point of view. So you start your experiments with molecules which are relatively simple for you,
I know,
but are quite complex for an astrochemist, right? So the question is, how do you think that the molecules that you are using as an input in your experiments are synthesized? So do you think that they are formed there from simpler precursors that maybe have arrived there from extraterrestrial delivery? And I'm asking this because in the recent years, we have been successful detecting some complexity in the interstellar medium, in the gas that would form stars and planets that, and in particular precursors of DNA. So these precursors that has been shown in prebiotic experiments by Sutherland group and Bonner group that can be used in prebiotic conditions to synthesize larger species, especially in the context of the RNA world. So, well,
I mean, nowadays,
about this.
I mean, the typical tough thing is the delivery on the Earth, and many people argue it's really tough because you just destroy these guys. There might be more ideas how ions could stabilize that, and Phil Schmidt-Coplin did a number of experiments that in our network, who knows, right? But I think it's a little bit always to think that the Earth had nothing, right? And it had to come from the outside, while these bodies which are bringing it in were also, you know, somewhere and produced apparently these molecules. It's kind of also indicating that the Earth was capable. Now, you know, which scenarios to get to these purines, if that's correct, or what you're going to go there. I mean, what John Sutherland is doing is very informative because you really get a lot of networks and a lot of dynamics. And for me, it's at this point a little bit hard to get that into that single pot experiment, really into it. And we'd like to dig out some older chemistries, but it might, as indicated and argued all the time at John Sutherland, not be very successful because the yield is very low if you want to do direct links and things like that. Now, it could be the accumulation in such a setting can help you there. And of course, if you would in situ also synthesize the molecules in its chiral forms in an equilibrium, you know, that could help you to push and purify one chirality more. But that's a bit, that's why I'm more and more, well, let's have a look. We explore a little bit that direction, how to get the G in the first place, but have not yet a good working hypothesis. The hope would be that polymerizing conditions and phosphorylating conditions could also help there somehow, but that's completely unclear at this point. But of course, that would be great.
Okay, thank you.
Thank you. Thank you very much. Are there any other questions? No? Let me see if there's anything else in the chat.
How will low gravity influence the efficiency of the chambers? You know, typically, if you make these chambers larger, right, our convection is too fast. So we would be happy for low gravity because then we could make flatter gradients to make the same effect. You know, the convection speed scales quadratically with the size, with the thickness of the chamber, and also diffusion scales quadratically because it equilibrates quadratically slower. So it, you know, it's easy to accommodate other gravities by just making the pore size a bit larger. It would actually help to have a little bit lower gravity.
Okay. Okay. Well, Thank you very much, Dita. I'm really sorry for the technical hiccups.
No, it's okay. No problem.
This hybrid mode is that's what happens.
We just have to learn it. And
yeah, maybe next
we'll
time you can come in person. Then we can have a normal seminar. So thanks so much for being with us today.
Yeah.
And I hope the best luck for this new renewal of this grant for the Origins of this initiative in Munich. Let's hope it is successful.
And let's hope that within Europe we'll manage to persuade people to push the
yeah,
field.
exactly.
Right.
Yes, that would be great.
Yeah.
And well, thanks so much. And we will see you all next week. Thank you, Dita.
Have a good one. Yes,
Take
thanks.
care. Bye-bye.
Bye-bye.